There's a fish that can see through its own head. Not metaphorically, literally. Right now, 2,000 feet beneath the Pacific, a creature hovers in absolute darkness with eyes embedded in a transparent skull. Tubular eyes, green, glowing, rotating inside a dome of clear tissue like organic telescopes scanning for prey in a world where sight shouldn't be possible. This isn't science fiction, it's the barreleye fish, and it's just the beginning. Because the ocean doesn't follow the rules we wrote for life. Down there in the crushing pressure and eternal night, evolution took a different path. A stranger path. Creatures exist that shouldn't be able to, with transparent bodies, bioluminescent organs, eyes the size of dinner plates on bodies 6 inches long, blood that's blue instead of red, metabolism so slow they can survive for months without food. They don't just look alien, they are alien. Their chemistry is different, their physics is different. The pressure that would crush a submarine is the pressure they need to stay alive. The darkness we fear is the light they avoid. The oxygen-depleted water that would suffocate us in seconds is the air they breathe. These aren't Earth creatures that adapted to the deep sea. These are deep-sea creatures that happen to share our planet. Tonight, we're diving into a world that breaks every biological rule we thought was absolute. Where evolution didn't just adapt, it invented. Where life found ways to exist that we're only beginning to understand. This is Omni. Stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture a moment to 500 feet below the surface. No light, no warmth, pressure so intense it would collapse your lungs in seconds. And in this darkness, a creature hangs motionless, waiting. It's the barreleye fish, Macropinna microstoma, and everything about it is wrong. Its head is a clear dome, not skin, not scales. A transparent shield filled with fluid, and inside that shield, 2 bright green cylinders point upward. Those are its eyes, not where you'd expect eyes to be, those dark spots on its face and nostrils. No, this fish's actual eyes are behind its face, embedded in its skull, free to rotate like a submarine's periscope. For 70 years after scientists first netted a barreleye in 1939, we thought we understood it. We thought those tubular eyes were fixed, locked in an upward gaze, giving the fish only tunnel vision of whatever drifted overhead. We were wrong. In 2009, researchers at the Monterey Bay Aquarium Research Institute did something remarkable. They brought a living barreleye to the surface and kept it alive long enough to watch. And what they saw changed everything. The eyes rotated. When the fish tilted its body from horizontal to vertical, the eyes turned with surgical precision, shifting from looking up to looking forward. According to marine biologist Bruce Robertson, who led the study, this transparent dome isn't just protection, it's a lens. It allows the fish to scan the water above for the faint silhouettes of prey, then rotate its eyes forward to actually catch and eat what it finds. But here's what makes this truly alien. The barreleye doesn't hunt in the open water. It hovers beneath massive jellyfish-like creatures called siphonophores, colonial organisms that trail stinging tentacles up to 130 feet long, like living drift nets. The barreleye carefully maneuvers through these forests of poison, stealing food that other animals have been caught and paralyzed. Its transparent shield protects those delicate rotating eyes from the stinging cells that would blind anything else. Think about what that means. This fish evolved a see-through head, rotating telescope eyes with bright green filters to distinguish sunlight from bioluminescence, and the spatial awareness to navigate through a maze of invisible, venomous tentacles in pitch-black water, all to steal someone else's lunch. That's not adaptation. That's invention. And the barreleye isn't alone. The deep ocean is filled with creatures whose very existence challenges what we thought life required. 3,000 feet down, in what was so depleted of oxygen that most fish would suffocate, the vampire squid thrives. Vampiroteuthis infinalis, literally vampire squid from hell, but it doesn't suck blood. It doesn't hunt. It drifts through oxygen minimum zones where the concentration of dissolved oxygen drops below 20 micromolar, less than 2% of what surface waters contain. To put that in perspective, you'd pass out in seconds if your blood oxygen dropped that low. Most ocean predators can't even enter these zones, but the vampire squid calls it home. How? Its blood is blue. Not red. It uses hemocyanin instead of hemoglobin to transport oxygen, and that copper-based molecule binds to oxygen with such efficiency that the vampire squid can extract every last molecule from water that would leave other creatures gasping. Its gills have evolved massive surface area. Its metabolism runs slower than any other cephalopods. It moves like it's conserving energy for a marathon that never ends. And when threatened, because even in the oxygen minimum zone predators exist, the vampire squid doesn't use ink. It has no ink sac. Instead, it expels a cloud of bioluminescent mucus, a sticky glowing mist filled with thousands of blue lights that pulse and shimmer in the darkness. According to research from the Monterey Bay Aquarium Research Institute in 2012, this luminous cloud can last nearly 10 minutes, and it serves two purposes. It blinds the predator with sudden light in a world of total darkness, and it sticks to the predator's body, making them glow, turning the hunter into a beacon for anything larger lurking nearby. The vampire squid has also evolved another defense. When disturbed, it inverts itself, turning inside out, wrapping its webbed arms up and over its body to expose rows of fleshy spines called cirri. It transforms from a soft, vulnerable creature into something that looks like a spiky alien pumpkin. And if that's not enough, it can turn off every bioluminescent organ on its body at will, becoming completely invisible in the dark. But here's what's truly remarkable. Unlike most cephalopods that live for 1 to 3 years and reproduce once before dying, vampire squids may live up to a decade. Female vampire squids can reproduce 20 times or more throughout their lifetime. They've evolved not just to survive in an environment that shouldn't support complex life. They've evolved to thrive there, slowly, patiently, across lifespans that dwarf their shallow water cousins. This is life playing by different rules, not bending the rules, rewriting them. And it's not just a few bizarre outliers, it's systemic. It's everywhere you look below a certain depth. According to a 2017 study published in Scientific Reports, researchers analysed over 350,000 observations from remotely operated vehicles, surveying the California coast from the surface down to 3,900 metres. They found that 76% of all observed organisms had bioluminescence capability, not 76% of species, 76% of individual creatures they encountered. In some parts of the ocean, particularly below 1,500 feet, that number climbs to nearly 90%. 9 out of 10 creatures you'd meet can create their own light through chemical reactions. It's not a rare adaptation, it's the dominant trait. Bioluminescence has evolved independently at least 40 separate times in marine organisms, 40 different. Lineages of life looked at the same problem: how do you see, communicate, hunt or hide in absolute darkness, and arrived at the same solution: make your own light. But they didn't all do it the same way. Some use bacterial symbionts. They carry colonies of glowing bacteria in specialised light organs. Others produce the light themselves through luciferin-luciferous reactions. Some glow blue-green because those wavelengths travel farthest through water. Others glow red, a colour most deep-sea creatures can't even see, giving them a private channel of communication and a hunting advantage like infrared night vision. The dragonfish, for instance, has evolved red bioluminescence and the rare ability to detect it. According to research published in Science in 2005, these fish essentially have built-in night vision goggles, allowing them to see prey that remain invisible to everyone else. This isn't just evolution fine-tuning traits. This is biochemistry inventing solutions to problems that don't exist on land. This is life under pressure, literal crushing, bone-shattering pressure, discovering that the laws of biology we learned in surface ecosystems don't apply down here. At 10,000 feet, the pressure reaches 300 atmospheres. That's 4,400 pounds per square inch. Imagine the entire NFL and NBA standing on your chest. That kind of force should collapse cells, denature proteins, crush membranes. And yet, life found a way. Deep-sea creatures have evolved proteins that maintain their shape under pressure. They've developed cell membranes with unique lipid compositions that stay fluid when they should be rigid. Some fish, like the Hadal snailfish found in the Mariana Trench at depths exceeding 26,000 feet, have evolved gelatinous, almost boneless bodies. Their flesh is so soft, so water-saturated, that when brought to the surface they lose all structure. According to a 2023 study published in eLife, these snailfish have lost genes for pigmentation, skeletal calcification, and even some visual wavelengths. They've shed every adaptation that doesn't serve them in the deepest trenches on Earth. What remains is streamlined for one thing: surviving where nothing else can. But it gets stranger. Some deep-sea creatures don't just survive the pressure, they die without it. Researchers have tried bringing deep-sea bacteria to the surface. Some species, collected from more than 6 miles down in the Mariana Trench, where pressure exceeds 1,000 atmospheres, simply won't grow at normal surface pressure. They've evolved so completely for the deep that our world is as hostile to them as theirs is to us. Think about that. There are organisms on this planet for whom our environment, the surface, with its breathable air and gentle pressure and sunlight, is lethal. We've created 2 Earths on one planet, and the one below the waves is larger. The ocean covers 71% of Earth's surface. The average depth is 12,100 feet. By volume, the deep sea, everything below 3,300 feet, is the largest living space on the planet. It's not an edge case, it's not a marginal habitat, it's the dominant ecosystem, and almost everything living there has had to become something we wouldn't recognize as possible. There are fish with antifreeze proteins in their blood to survive near freezing temperatures. There are worms that live in toxic sulfide plumes from underwater volcanoes, farming bacteria that convert poison into food. There are jellies made of 99% water that drift through the water column, with such minimal body structure they're barely more than organized liquid. There's the giant isopod, a creature that looks like a pill bug the size of a football, with a metabolism so slow it can go 5 years without eating. There's the gulper eel with a mouth larger than its entire body, hinged jaws that can unhinge to swallow prey twice its size. There's the anglerfish, where the male, a tiny parasite, fuses permanently to the female's body, dissolving until only his reproductive organs remain, a living sperm bank grafted to her flesh. These aren't just strange animals, these are fundamentally different solutions to the problem of existence. On land, life evolved within a narrow band of conditions: temperature, pressure, oxygen, light. They all stay relatively stable. Evolution on land is about competing for resources within those constraints. But in the ocean, especially the deep ocean, those constraints don't exist. Or they exist in such extreme forms that life had to invent entirely new metabolic pathways, entirely new structural chemistry, entirely new sensory systems. And here's what we're only beginning to understand. These creatures aren't poorly adapted surface animals struggling to survive in a harsh environment. They're perfectly adapted deep-sea organisms for whom the surface would be a nightmare. The pressure we fear, they need it. The darkness we die in, it's their refuge. The cold, the scarcity, the isolation, those aren't obstacles. Those are features of the ecosystem they've mastered. We've been thinking about deep-sea life all wrong. We've been asking how do they survive down there? When we should be asking, what did the ocean allow life to become that the land never could? Because the ocean didn't just hide creatures from another world, it created another world. A world where evolution followed different physics, different chemistry, different biological logic. And tonight we're going to trace how it happened. How pressure reshaped cells, how darkness birthed light, how scarcity engineered patience, how isolation fostered the strangest biodiversity on Earth. We'll explore the chemistry that shouldn't work, the anatomy that defies structure, the senses that detect what can't be seen, the metabolisms that run on almost nothing. By the end, you might not look at the ocean the same way, because it's not just water, it's a laboratory, and for 400 million years, it's been running the most radical experiment in evolutionary biology Earth has ever seen. Let's begin with the simplest question, the one that breaks everything we thought we knew about life. How do you see in a place where light doesn't exist? The question seems simple. How do you see in a place where light doesn't exist? The answer evolution found is almost paradoxical. You make your own. Bioluminescence, the production of light by living organisms through chemical reactions, isn't rare in the ocean. It's not even uncommon. It's the dominant form of communication on the planet. More organisms speak in light than in sound, in scent, in touch. In the mesopelagic zone, the twilight layer between 600 and 3,300 feet deep, an estimated 76% of all visible organisms can produce their own light. Let that sink in. 3 out of every 4 creatures you'd encounter in the largest habitat on Earth glow. But here's what makes this extraordinary. Bioluminescence didn't evolve once and spread. It evolved independently, over and over again, in completely unrelated organisms solving the same problem in the same way. According to research published across multiple studies, bioluminescence has arisen at least 40 separate times across the tree of life, and likely more than 50 times when you account for recent discoveries. In 2016, researchers analysing ray-finned fishes alone found that bioluminescence evolved 27 independent times just within that group, 27 separate occasions where fish lineages, scattered across millions of years of evolution, looked at the darkness and invented light. This is convergent evolution on a scale that borders on inevitability. When the same solution appears dozens of times independently, you're no longer looking at accident. You're looking at physics and chemistry conspiring to make certain outcomes nearly mandatory. The chemistry itself is elegant. At its core, bioluminescence requires a light-emitting molecule called luciferin and an enzyme called luciferase. When luciferin reacts with oxygen in the presence of luciferase, it releases energy, not as heat, but as cold light. It's one of the most efficient energy conversions in nature, with nearly 100% of the chemical energy transformed directly into photons. But here's where it gets strange. Luciferin doesn't vary much across species. One molecule in particular, calentrazine, serves as the light-emitting pigment for 9 completely different phyla. Radiolarians, comb jellies, cnidarians, crustaceans, mollusks, arrowworms and vertebrates all use the same basic chemical to glow. Yet luciferase, the enzyme that controls the reaction, varies wildly. Almost every species has its own unique version. It's as if evolution handed out the same flashlight bulb to dozens of unrelated organisms, but each one had to invent their own battery and switch. Some organisms synthesize calentrazine themselves, others obtain it through their diet, eating creatures that produce it and recycling the molecule for their own uses. It creates a glowing food web where light compounds cascade through the ecosystem like nutrients, passed from prey to predator, each organism adding its own bioluminescent signature. And the purposes? They're as varied as the organisms themselves. Some use light to hunt. The anglerfish is famous for this, a glowing lure dangling from a modified dorsal spine, wiggling in the darkness like bait on a fishing line. Prey sees movement, approaches what they think is food, and find teeth instead. The anglerfish doesn't chase. It waits, motionless in the black water, letting curiosity do the work. But not all anglerfish lures work the same way. Different species have evolved wildly different light organs. Some have simple glowing bulbs. Others have elaborate branching structures filled with bioluminescent bacteria housed in specialized chambers. The flashlight fish, relatives of the anglerfish, have pockets beneath their eyes filled with luminous bacteria that they can cover and uncover with muscular shutters, creating blinking patterns like Morse code. According to a 2016 study in PLOS One, these bacterial partnerships have evolved at least 17 separate times in fish alone. The bacteria, mostly from the genus Vibrio, are essentially free-living organisms that fish capture from the environment and cultivate in specialized organs. The fish provide nutrients and protection, the bacteria provide light, it's symbiosis written in photons, and the fish control that light with remarkable precision. Ponyfishes, for instance, have evolved sexually dimorphic shutters and species-specific translucent windows to focus or broadcast bacterial light in patterns unique to each species. Deep-sea anglerfishes have sculpted their bacterial light organs into elaborate lures that vary so dramatically between species that you can identify them by their eskers alone. Other creatures use light for camouflage, which sounds counterintuitive until you understand the physics. In the twilight zone, faint sunlight still filters down from above. If you're a fish swimming in mid-water, you cast a silhouette, a dark shape against the dim glow from the surface. Predators lurking below can see that silhouette clearly. But if you have photophores, light-producing organ, on your underside that match the intensity and colour of the downwelling light, you erase your shadow. You become invisible. This is called counter-illumination, and it's so effective that dozens of species have independently evolved the same strategy. The hatchetfish, for example, has rows of photophores along its belly that it continuously adjusts to match the ambient light from above. Photoreceptors on its back measure incoming light. Neural circuits calculate the needed intensity. The photophores respond in real time, dimming and brightening to maintain perfect camouflage. It's active camouflage controlled by what is essentially a biological light meter and feedback loop, operating automatically in a fish brain the size of a grain of rice. Then there's defensive bioluminescence, and this is where things get creative. When attacked, some organisms don't run. They scream, not in sound, in light. They produce what researchers call a burglar alarm display, a sudden explosion of bioluminescence designed not to blind the predator, but to attract something bigger. The strategy is brilliant. If you're being eaten, light up as brightly as possible. Make yourself and your attacker visible. Anything larger and hungrier that sees the commotion will investigate, and suddenly your predator has become prey. You, in the chaos, slip away into the darkness. The vampire squid does a variation of this. When threatened, it ejects that sticky cloud of bioluminescent mucus, thousands of glowing particles that adhere to the predator's body, marking them like a beacon. According to observations from the Monterey Bay Aquarium Research Institute, this luminous cloud can persist for nearly 10 minutes, plenty of time for larger predators to home in on the glowing target. But perhaps the most sophisticated use of bioluminescence is communication, especially in finding mates. In the vast, dark ocean, how do you find another member of your species? Sound doesn't travel well at certain frequencies. Chemical signals disperse slowly. But light? Light can be seen from hundreds of feet away, and it can encode complex information in ways scent never could. Many bioluminescent species have evolved species-specific flash patterns, rhythms and intensities unique to each type. Firefly squid, for instance, gather in massive mating aggregations off the coast of Japan each spring, creating synchronized light shows that can be seen from shore. Each species flashes its own pattern, a visual dialect that ensures they only attract the right mates. Lanternfishes, the family Myctophidae with over 250 species, are perhaps the most diverse bioluminescent vertebrates. They're also among the most abundant fish in the ocean, and each species has a unique arrangement of photophores on its body. Pattern and position matter. Number of photophores matters. Sexual dimorphism exists. Males and females often have different light arrangements. According to a 2024 study in Nature Ecology and Evolution, this diversity isn't accidental. Lanternfishes that evolved species-specific bioluminescent patterns diversified into new species at much faster rates than their relatives without them. The light patterns act as reproductive isolators. Fish that recognize each other's flash patterns mate preferentially, splitting populations even when they share the same habitat. Bioluminescence, in this case, doesn't just help fish find mates, it actively drives speciation. It's both a communication system and an evolutionary engine. And then there's red bioluminescence, which is vanishingly rare and utterly fascinating. Most deep-sea bioluminescence is blue-green. Blue wavelengths travel farthest through water, up to 100 meters in clear ocean water, compared to just 15 meters for red light. Red light is absorbed so quickly that by the time you're 50 feet deep, it's functionally extinct. This is why many deep-sea animals are red. In the absence of red light, red pigmentation renders them invisible, appearing black against the dark water. But a handful of species, notably dragonfish in the family Stomidae, have evolved the ability to produce and see red bioluminescence. According to research published in Science in 2005, these fish have a secret spotlight. They produce red light from photophores beneath their eyes, illuminating prey that other predators can't detect. It's like having infrared night vision in a world where everyone else is blind to that spectrum. They can see their food can't see them. Their competitors can't see what they're doing. The chemistry required for this is extraordinary. Producing red bioluminescence requires a different luciferin or modifications to the standard blue-green systems. The visual pigments needed to detect red light require specialized opsins tuned to longer wavelengths. These fish had to evolve both the sender and the receiver, the flashlight and the eyes to see by it. And they did it independently. At least 3 separate lineages of dragonfish evolved red bioluminescence, each using slightly different chemical mechanisms. Convergent evolution again, but this time on a private channel. What does all this tell us? That bioluminescence isn't an exotic adaptation. It's a fundamental reimagining of what light means. On land, light comes from the sun. In the deep ocean, light comes from life. It's not just vision. It's language, camouflage, weaponry, courtship, speciation. It's chemistry turned into communication, at scales we're only beginning to measure. But it raises a deeper question. If the absence of light drove the evolution of bioluminescence 40+ independent times, what else has the deep ocean forced life to reinvent? Because light is just the beginning. The darkness is only one challenge. There's also the pressure, and the pressure changes everything. At 10,000 feet below the surface, the pressure reaches 300 atmospheres. That's 4,400 pounds per square inch pressing down on every surface. To put that in human terms, imagine the entire NFL and NBA standing on your chest, all at once. That's what a fish experiences just existing at that depth. At 26,000 feet, the depth of the Mariana Trench, pressure exceeds 1,000 atmospheres, over 15,000 pounds per square inch. At this pressure, styrofoam cups shrink to the size of thimbles, air pockets in submersibles compress to dangerous levels. The human body, being mostly water, would fare slightly better than a submersible, but our insistence on keeping air in our lungs would kill us instantly. And yet, life exists there, not struggling, thriving. The Hadal snailfish, Pseudolaparus swirri, holds the record for the deepest living fish ever captured. Found at 26,131 feet in the Mariana Trench, this pale, translucent creature swims effortlessly in water that would crush almost anything from the surface. It has no scales, no swim bladder. Its bones are barely calcified, giving it a gelatinous, almost boneless structure. But here's the truly remarkable part. When brought to the surface, these fish die. Not from explosive decompression like you'd expect, but because their biochemistry literally stops working at normal pressure. They're not adapted to survive the deep. They're evolved to require it. The key to understanding this lies in a molecule with a distinctly unpleasant association, trimethylamine oxide, or TMAO. If you've ever smelled fish that's gone bad, you've smelled trimethylamine, the breakdown product of TMAO that gives rotting seafood its characteristic stench. But in living fish, TMAO serves a far more elegant purpose. It's a pressure counteractant, a molecular shield that protects proteins from being crushed and deformed by the immense forces of the deep. The story of TMAO begins with a simple observation. In the early 2000s, marine biologist Buoyancy at Whitman College noticed something strange. Shallow water fish typically have TMAO concentrations around 40-70 millimoles per kilogram of muscle tissue. But when his team analysed deep-sea fish, grenadiers, rat-tails and other species living at 60 to 16,000 feet, the TMAO levels were dramatically higher. According to research Yancey published in 2014 in the Proceedings of the National Academy of Sciences, the pattern was striking. TMAO concentration increased linearly with depth. A fish at 3,000 feet might have 100 mg, at 6,000 feet 150 mol, at 13,000 feet over 200 mol, and the Hadal snailfish from 26,000 feet, 261 mol, the highest TMAO concentration ever recorded in any fish. This wasn't random. TMAO levels tracked depth with mathematical precision, increasing at roughly 20 millimoles per kilometre of descent. And of all the environmental variables that change with depth, temperature, light, oxygen, nutrients, only one changes in a perfectly linear way: pressure. But why would fish need more TMAO as pressure increases? The answer lies in what pressure does to proteins. Proteins are chains of amino acids folded into precise three-dimensional shapes. Those shapes determine function. An enzyme works because its active site has exactly the right geometry to bind substrates and catalyze reactions. A structural protein provides support because its folded configuration creates rigidity. Pressure destabilizes these folds. At high pressure, the forces between atoms shift. Hydrogen bonds weaken. Hydrophobic regions that normally cluster together spread apart. The protein begins to denature, not from heat, but from mechanical compression. For most proteins from surface organisms, pressures above 400 to 500 atmospheres cause catastrophic loss of function. Enzymes stop catalyzing. Structural proteins collapse. Muscle fibers fail to contract. Life, as designed for the surface, simply breaks. But TMAO changes the game. According to studies conducted by Buoyancy and marine biochemist Joseph Siebenhaler, TMAO acts as what's called a chemical chaperone. It doesn't bind directly to proteins. Instead, it alters the properties of the water surrounding them. Water under pressure becomes more ordered, more structured. This structured water penetrates into proteins, forcing open cavities and unfolding regions that should stay compact. But TMAO disrupts that process. It bonds with water molecules in ways that preserve the water's normal, less structured state, even under crushing pressure. The result? Proteins stay folded, enzymes keep working, muscle contracts, life continues. In laboratory experiments, researchers subjected lactate dehydrogenase, a critical enzyme in muscle metabolism, to 500 atmospheres of pressure. Without TMAO, the enzyme's activity dropped by 15%. Its ability to bind substrates degraded. At even higher pressures, it lost half its function. But when 250 millimoles of TMAO was added to the solution, the enzyme worked perfectly. At 500 atmospheres, equivalent to living at 16,000 feet deep, performance matched what you'd expect at the surface. The same held true for other proteins. Actin, the protein that forms muscle filaments, normally can't polymerize under high pressure. The pressure disrupts the assembly process, but add TMAO and actin polymerizes normally even at 500 atmospheres. According to a 2022 study published in Communications Chemistry, researchers used neutron scattering to observe exactly what TMAO does at the molecular level. They subjected water to 4,000 bar of pressure, roughly 40 times atmospheric pressure, both with and without TMAO present. Under pressure alone, the water's hydrogen bonding network collapsed. The second solvation shell, the layer of organized water molecules surrounding other water molecules, disappeared. This is the change that penetrates proteins and destroys their structure. But with TMAO present, that second solvation shell remained intact. TMAO formed strong hydrogen bonds with water molecules, creating a protective cage that resisted pressure-induced distortion. The water, even under extreme force, behaved almost as if it wasn't under pressure at all. And here's where it gets mathematically beautiful. The researchers calculated an osmolyte protection ratio, the concentration of TMAO needed to counteract a given amount of pressure. That ratio, derived from molecular physics, matched almost perfectly with the TMAO concentrations actually found in fish at corresponding depths. Physics predicts what evolution discovered, but there's a catch, a fundamental limit. Fish are osmoregulators, unlike invertebrates, which allow their internal osmolality to match seawater, roughly 100 milliosmoles per kilogram. Fish actively maintain lower internal osmolality, typically around 350 µm kilograms. They do this by excreting salt through their gills and kidney, maintaining an osmotic gradient that keeps water from constantly flooding their cells. But TMAO is an osmolyte. It contributes to internal osmotic pressure, and as TMAO concentrations rise with depth, so does the fish's total internal osmolality. At shallow depths, this isn't a problem. A fish with 50 molk TMAO maintains an osmolality well below seawater, but a Hadal snailfish with 261 molk TMAO pushes internal osmolality towards 700-800 mmol/kg. That's approaching the osmolality of seawater itself. Yancey's team did the math. If you extrapolate the linear increase of TMAO with depth, fish would reach isosmotic conditions, where their internal fluids match seawater, somewhere between 26,000 and 28,000 feet. Beyond that depth, fish would need more osmolality than seawater to continue producing enough TMAO. They'd have to reverse their osmotic gradient. salt would flow inward instead of outward. Their entire osmoregulatory system, evolved over hundreds of millions of years to pump salt out, would have to run backward. It's mechanically possible, but evolutionarily implausible. The energetic costs would be staggering. The cellular machinery would need complete redesign, and tellingly, no fish have ever been found below 27,000 feet. The deepest 25% of the ocean, from 27,000 to 36,000 feet, appears to be entirely fishless. This isn't speculation. Multiple deep-sea surveys using baited cameras, submersibles and traps have explored Hadal trenches worldwide. They find amphipods, sea cucumbers, snailfish down to about 27,000 feet. Below that, invertebrates continue, but fish disappear. TMAO may be the biochemical barrier that sets the depth limit for all fish. Not because pressure crushes them, but because the chemistry required to resist that pressure creates osmotic constraints they can't overcome. Life isn't prevented from going deeper by mechanical forces, it's constrained by the math of molecular protection. But this raises an even stranger question. If fish are biochemically locked out of the deepest trenches, what about organisms that don't have the same osmotic constraints? Invertebrates, particularly amphipods, have been found thriving at 36,000 feet, the very bottom of the Mariana Trench, and they use the same TMAO strategy. According to a 2018 study published in Deep Sea Research, amphipods from Hadal depths contain TMAO concentrations exceeding 300 mmol and even higher than the deepest fish. But amphipods are osmoconformers. They don't fight against seawater's osmolality, they match it. Their internal fluids are naturally around 100 massamosmkus and dominated by sodium and chloride. Adding TMAO doesn't create an osmotic problem because they're not trying to maintain a gradient. They can keep increasing TMAO indefinitely without hitting the wall fish encounter. So the deepest parts of the ocean aren't lifeless, they're just reserved for organisms with different osmotic strategies. Fish evolved in a way that brilliantly solved mid-depth survival, but accidentally created a ceiling they can't break through. Invertebrates following a different evolutionary path face no such limit. It's a stunning example of how evolution's solutions aren't perfect. They're constrained by prior commitments. Fish became osmoregulators hundreds of millions of years ago, and that decision, made in shallow seas, ultimately determines how deep their descendants can ever go. But TMAO isn't the only pressure adaptation, it's just the most elegant chemical one. Other organisms have found different solutions. Some bacteria from Hadal depths have evolved pressure-activated enzymes, proteins that actually work better under compression. Their optimal activity occurs at 400–800 atmospheres. At surface pressure, they're sluggish, inefficient. Take them to the deep, and they thrive. According to research on pisophilic, pressure-loving bacteria published in Frontiers in Microbiology, some species have even evolved pressure-dependent gene regulation. Certain genes only activate above 200 atmospheres. They've wired pressure into their genetic circuitry as an environmental signal, the way surface organisms use temperature or light. And then there are the structural adaptations that seem almost paradoxical. Deep-sea fish often have gelatinous, watery tissues, up to 95% water in some species. Their bones are poorly mineralized, sometimes barely present. They have minimal musculature. This isn't weakness, it's strategy. Water is incompressible. Unlike air, which collapses under pressure, water maintains its volume. A body filled with water doesn't get crushed because there's nothing to compress. The pressure outside equals the pressure inside and everything equilibrates. But proteins, lipids, and complex molecules can still deform, so deep-sea organisms maximize water content and minimize the structural components that pressure can damage. They become, essentially, organized bags of seawater with just enough biochemistry to stay alive. The blobfish, made famous by photos showing its gelatinous, sad-looking face, is the perfect example. At depth, under pressure, the blobfish looks relatively normal. Its tissues, filled with low-density gel, maintain structure because the internal and external pressures balance. But bring it to the surface and it collapses. The pressure that was supporting its tissues vanishes. What was once firm at 3,000 feet becomes a shapeless blob at sea level. The blobfish didn't evolve to look sad. It evolved to look normal at depth, and we only see it out of context. Similarly, many deep-sea creatures have lost swim bladders. gas-filled organs that provide buoyancy in shallow fish. A swim bladder at 10,000 feet would compress to a fraction of its normal size, becoming useless or even dangerous. So deep-sea fish evolved different buoyancy strategies: oily livers, gelatinous tissues, reduced skeletal mass. They achieve neutral buoyancy through chemistry, not pneumatics. And their movement strategies change too. Muscle requires energy. At great depths, where food is scarce and metabolism runs slow, fast swimming is prohibitively expensive. So many deep-sea predators evolved ambush strategies. They hover, they drift, they wait with minimal movement, conserving every calorie until prey wanders close enough to strike. The gulper eel exemplifies this. Its body is essentially a giant mouth attached to a thin whip-like tail. It can unhinge its jaws to swallow prey larger than itself, and its stomach expands to accommodate huge meals. Because food is so rare at 6,000 feet, it has evolved to eat anything it encounters, regardless of size, then digest slowly over weeks. This is pressure's second effect. It doesn't just crush, it isolates. Food sinks slowly, nutrients disperse. The deep ocean is a desert, and pressure is part of why. Pressure makes it energetically expensive to move, to hunt, to maintain fast metabolisms, so life slows down, and in slowing down it becomes patient in ways surface organisms never achieve. The Greenland shark, which hunts in deep, cold Arctic waters, at pressures exceeding 200 atmospheres, has a metabolism so slow that it grows less than a centimetre per year. It doesn't reach sexual maturity until age 150. Some individuals may live over 400 years, the longest-lived vertebrates on Earth. That's not just adaptation to cold, it's adaptation to an entire suite of deep-sea conditions where pressure, temperature, darkness, and scarcity conspire to make speed and aggression wasteful. Patience, in the deep, is survival. What does all this mean? It means pressure didn't just shape what organisms look like, it shaped how they think, how they move, how they allocate energy, how fast they live. It created biochemical barriers that entire groups can't cross. It forced life to invent molecules like TMAO that we're only now understanding at the atomic level. And it reveals something profound. The deep ocean isn't Earth with more water on top. It's a fundamentally different physical regime, where the rules of biochemistry, physiology, and ecology all had to be rewritten. But pressure is only half the equation. There's something else the deep ocean lacks. Something surface life takes for granted. Something so fundamental that its absence should make complex life impossible. Oxygen. You could drown in oxygen-rich water. Your lungs would fill, and you'd suffocate. But imagine drowning in water that itself is suffocating. Water so depleted of oxygen that even fish can't breathe. These zones exist. They're called oxygen minimum zones, or OMZs, and they occupy massive volumes of the ocean, primarily at depths between 600 and 3,000 feet. In some regions, off the coast of California, in the eastern tropical Pacific, the Arabian Sea, oxygen concentrations drop to less than 0.5% of surface levels. To put that in perspective, surface seawater at equilibrium with the atmosphere contains roughly 200 250 micromoles of oxygen per litre. In oxygen, minimum zones, concentrations can plummet below 20 micromoles per litre. In the most extreme cases, in what researchers now call anoxic marine zones, oxygen becomes undetectable even with the most sensitive instruments. Less than 5 micromoles per litre, essentially zero. And yet, life exists there, not barely clinging to survival, thriving, abundant. Some of the ocean's richest ecosystems exist in these suffocating layers. The question isn't whether life can survive low oxygen. The question is how it's possible at all. The answer involves some of the most radical biochemical engineering evolution has ever produced. First, understand what oxygen minimum zones are and why they form. They're not random. They're predictable consequences of ocean physics and biology working together. Start at the surface. Sunlight drives photosynthesis. Phytoplankton, microscopic algae drifting in the water column, produce oxygen as a byproduct. Surface waters stay well oxygenated, constantly mixing with the atmosphere, saturated with dissolved gases. But phytoplankton don't live forever. They die, they sink, and as they sink, they become food. Bacteria in the water column feast on this marine snow, a a constant rain of dead plankton, fecal pellets, shed scales, mucus, organic debris. And bacteria breathe. They consume oxygen to metabolize organic matter, breaking it down into nutrients and carbon dioxide. The deeper you go, the more accumulated detritus there is to consume, and crucially, the less mixing occurs with oxygen-rich surface water. Deep water moves slowly, sometimes taking decades to circulate back to the surface. Once the oxygen in that water is consumed by microbial respiration, it can't be replaced without the water mass reaching the surface again. In certain regions, particularly where nutrient-rich upwelling drives massive phytoplankton blooms, creating huge amounts of sinking organic matter, microbial respiration consumes oxygen faster than circulation can replenish it. The result, an oxygen minimum zone, a layer of water, often hundreds of meters thick, where dissolved oxygen drops to levels that would kill most surface-adapted organisms in minutes. According to a study published in the Journal of Experimental Biology in 1998 by marine physiologists James Childress and Brad Seibel, animals living in oxygen minimum zones off the coast of California face partial pressures as low as 0.8 kilopascals, about 6 torr, or less than 1% of atmospheric pressure. To a mammal, that's lethal. You'd lose consciousness within seconds. But the animals there have solved this problem through a suite of adaptations so complete, so integrated, that they've essentially re-engineered how respiration works. Start with extraction efficiency. Most fish extract about 50–60% of the oxygen from water, passing over their gills. That's respectable. But in oxygen minimum zones, that's nowhere near enough. Species like the bathypelagic mysid Gnathopausia ingens, a shrimp-like crustacean that lives at depths where oxygen drops to 6 torr, can extract up to 90% of available oxygen from ventilated water. How? Massive gills with enormous surface area. According to research published in the Biological Bulletin in 1990 by Childress and colleagues, these animals have evolved gill structures with exceptionally thin diffusion barriers, sometimes just a few micrometers separating water from blood, and gill surface areas far exceeding what you'd find in surface-dwelling relatives. But extraction is only half the problem. You also need transport. This is where hemocyanin becomes critical. Most vertebrates use hemoglobin, an iron-based protein that turns blood red, to transport oxygen. Hemoglobin works brilliantly at normal oxygen levels, but many deep-sea invertebrates, including crustaceans and cephalopods, use hemocyanin instead, a copper-based protein that turns blood blue. And in oxygen minimum zones, hemocyanin has a crucial advantage. It can be tuned to have extraordinarily high oxygen affinity. Gnathopausia ingens has hemocyanin with a P50, the oxygen partial pressure at which the protein is 50% saturated, of just 1.4 torr at physiological temperature and pH. That means this animal's blood can load oxygen even when environmental levels drop to barely detectable amounts. For comparison, human hemoglobin has a P50 around 27 torr. At 6 torr, your hemoglobin would be less than 25% saturated. You'd be hypoxic, approaching organ failure. But Gnathopausia's hemocyanin would still be over 80% saturated, fully functional. But here's the engineering challenge. If your blood protein binds oxygen too tightly, it won't release it at the tissues. High affinity is useless if you can't deliver. Deep-sea animals solve this with cooperativity and the Bohr effect. Cooperativity means that once one oxygen molecule binds to hemocyanin, the protein changes shape slightly, making it easier for additional oxygen molecules to bind. This creates a steep binding curve. Oxygen loads rapidly when available, but unloads rapidly when partial pressure drops slightly. Gnathopausia's hemocyanin has a cooperativity coefficient, N50, of 3.5. Exceptionally high for a crustacean. This means that across a very small change in oxygen pressure, say from 6 torr in the gills to 4 torr in the tissues, the hemocyanin goes from nearly fully loaded to substantially unloaded, delivering oxygen efficiently despite the tiny gradient. And then there's the Bohr effect, the tendency for respiratory proteins to release oxygen when pH drops. In metabolically active tissues, cells produce carbon dioxide, which lowers local pH. This acidification causes hemocyanin to release oxygen right where it's needed most. Gnathopausia has one of the largest Bohr effects ever measured in a crustacean respiratory protein, a log P50 dpH of 0.80. That means a small drop in pH causes a massive increase in oxygen release. Put it all together. Massive gills extract 90% of available oxygen. High affinity hemocyanin loads that oxygen even at 1,2 torr. High cooperativity and a large Bohr effect ensure efficient delivery to tissues across minimal gradients. It's biochemical perfection, and it's not unique. The vampire squid, living in even more extreme oxygen minimum zones, where levels drop below 5 micromoles per litre, has evolved similar strategies. Its hemocyanin binds oxygen so efficiently that the animal can extract enough to support aerobic metabolism in water that would asphyxiate almost any other active predator. According to research published in Progress in Oceanography in 2010, the vampire squid's closest relatives, other omastrophid squids like Ilex illicabrosus, have hemocyanin P50 values around 8-10 torr. They're adapted to well-oxygenated Atlantic waters. But the vampire squid, its P50 drops below 2 torr, and remarkably, that affinity changes with temperature and depth. When the squid is in cold, deep, oxygen-depleted water, its hemocyanin affinity increases. When it occasionally ventures into warmer, oxygen-rich surface waters, affinity decreases, facilitating faster oxygen release to active tissues. It's dynamic regulation. The same protein functions differently depending on environmental conditions, tuned by temperature and pH to optimize performance across wildly varying oxygen landscapes. But even the most efficient oxygen extraction and transport can't support high activity levels when oxygen is scarce. So many oxygen minimum zone residents have adopted a different strategy: metabolic suppression. The Humboldt squid, Dosidicus gigas, is a massive active predator that hunts in the oxygen minimum zones of the eastern Pacific. These squid can reach 6 feet in length and weigh over 100 pounds. They're aggressive, fast-swimming carnivores with metabolic demands that should be incompatible with low-oxygen environments, and yet, they spend significant portions of their day in the oxygen minimum zone, at depths where oxygen concentrations drop below 10 micromoles per litre. How? According to studies conducted by Seibel and published in 2011 in the Journal of Experimental Biology, Humboldt squid suppress their metabolism by up to 40% when they enter low-oxygen waters. They don't shut down completely. They remain alert, capable of predation, but they dramatically reduce energy expenditure. Their routine metabolic rate, the energy needed just to stay alive and move slowly, drops from roughly 50 micromoles of oxygen consumed per gram per hour at the surface to around 30 micromoles per gram per hour in the oxygen minimum zone. This isn't passive drowsiness, it's active metabolic regulation. The squid reduce gill ventilation frequency. They slow movement. They may even reduce neural activity in non-essential systems. And they switch partially to anaerobic metabolism, producing lactate as a byproduct of glycolysis when aerobic ATP production can't meet demands. But anaerobic metabolism is expensive and unsustainable. Lactate accumulates. It creates acid. It has to be cleared eventually, and clearing it requires oxygen. So the Humboldt squid uses anaerobic pathways only as a supplement, covering maybe 10-20% of their energy needs during hypoxic forays. The rest comes from ruthlessly efficient aerobic metabolism running at minimal intensity. And here's the fascinating part. This strategy is reversible. When the Humboldt squid ascends at night into oxygen-rich surface waters to hunt, its metabolism ramps back up. Heart rate increases, ventilation frequency increases. It pays back the oxygen debt accumulated during the day, clearing lactate, restoring ATP reserves. The squid essentially commutes between two metabolic states, suppressed and aerobic in the daytime oxygen minimum zone, fully active and aerobic at night in surface waters. It's a daily metabolic migration as dramatic as its vertical migration through the water column. Other species don't migrate. They live permanently in the oxygen minimum zone, and for them, metabolic suppression isn't temporary, it's a way of life. Deep-sea fishes that inhabit oxygen minimum zones have routine metabolic rates 10 to 20% of what you'd find in similarly sized shallow water species. They move slowly, they grow slowly, they reproduce infrequently. Everything is paced to minimize oxygen consumption. But don't mistake this for weakness. These fish aren't struggling, they're optimized. In an environment where food is scarce and oxygen is limited, A slow metabolism is an advantage. It means longer survival between meals. It means less vulnerability to brief periods of even lower oxygen. It means energy can be allocated to reproduction, growth, and immune function, instead of wasted on unnecessary activity. And the evolutionary results are striking. According to a 2011 study by Seibel, organisms living permanently in oxygen minimum zones have critical oxygen levels, the picoret, below which they can no longer maintain aerobic metabolism, as low as 1 to 2 torr. Compare that to surface species, which typically have picoret values of 40-80 torr, and the adaptation becomes clear. An organism with a picoret of 2 torr can remain fully aerobic in water where oxygen is barely detectable. It's not gasping, it's not distressed, it's functioning normally in conditions that would be lethal to its ancestors. But there's a limit to even these adaptations. In the most extreme oxygen minimum zones, the anoxic marine zones where oxygen drops to 0, even the most efficient aerobic metabolism fails. And here, life faces a choice: leave or switch to chemistry that doesn't require oxygen at all. Some bacteria make that switch. They use nitrate instead of oxygen as an electron acceptor, conducting denitrification, a form of anaerobic respiration that converts nitrate to nitrogen gas. Vast bacterial communities thrive in anoxic marine zones, cycling nitrogen through pathways that bypass oxygen entirely. These microbes can dominate the ecosystem. In the oxygen minimum zone off the coast of South America, researchers have found bacterial mats the size of Uruguay floating in the water column, processing nutrients, consuming organic matter, and fundamentally altering ocean chemistry on a continental scale. But complex animals, fish, squid, crustaceans, can't sustain themselves on anaerobic metabolism alone. The energy yield is too low, the waste products too toxic. So the true anoxic zones, where oxygen reaches 0 and stays 0, are largely devoid of complex metazoans. They're bacterial worlds. Which brings us to a profound realization. Oxygen minimum zones aren't just challenging environments that a few hardy species tolerate. They're selective filters that have driven some of the most sophisticated respiratory and metabolic adaptations in the animal kingdom. The vampire squid's hemocyanin, the Humboldt squid's metabolic suppression, Gnathopasia's massive gills and cooperative oxygen binding. These aren't minor tweaks. They're comprehensive re-engineering of how energy is acquired and used. And they reveal something crucial. The deep ocean didn't just remove light and add pressure. It also removed the oxygen we take for granted. And in doing so, it forced life to invent respiratory strategies that don't exist on land, metabolic flexibilities surface organisms never needed, biochemical solutions to problems that never arose in well-oxygenated ecosystems. The ocean minimum zones are laboratories of physiological innovation, and the creatures that live there are proof that impossible conditions just mean evolution hasn't had enough time yet. But even metabolic suppression, high affinity hemocyanin, and massive gills can't explain everything we see in the deep. Because some adaptations aren't about extracting more from less. They're about sensing what shouldn't be sensible, detecting signals in a world where conventional senses fail. In the darkness and scarcity of the deep, survival isn't just about breathing. It's about knowing what's out there, even when you can't see, can't hear, and can barely detect anything at all. In complete darkness, under crushing pressure, with minimal oxygen and almost no sound, how do you find food? How do you avoid becoming food? How do you find a mate in three-dimensional space when you can't see more than a few feet in any direction? The answer: you stop relying on the senses that work on land, and you develop entirely new ways of perceiving the world. Deep-sea creatures don't just have better versions of human senses, they have senses we don't possess at all. Senses that detect things we can't even imagine detecting. Senses that turn the ocean into a readable landscape of information invisible to us. Start with the lateral line system, perhaps the most alien sensory organ fish possess, and one we have no equivalent for. Every fish, from shallow water minnows to hadal-zone snailfish, has a lateral line running along each side of its body. If you look closely at a fish, you'll see it, a faint line of small pores running from head to tail. Those pores aren't decorative. They're openings to a sophisticated mechanoreceptive network that allows fish to feel water movement at a distance. Inside those canals, embedded in the skin, are specialized sensory structures called neuromasts. Each neuromast consists of hair cells, the same type of mechanoreceptive cells found in your inner ear, topped with a gelatinous cupula that acts like a fluid-filled dome. When water moves across the fish's body, it creates pressure differentials. Water flows through the pores, creating currents in the lateral line canals. Those currents bend the cupulae, which deflect the hair cells beneath them. The hair cells depolarize, sending electrical signals to the brain. The result? The fish can detect vibrations, pressure waves, and water displacement from meters away. It can sense the movement of nearby objects without seeing, hearing, or touching them. It can feel the wake left by another fish that passed by seconds ago, tracking prey or predators through invisible trails in the water. According to research on the lateral line system, fish use this sense for everything: navigating in darkness, maintaining position in schools, detecting obstacles, locating prey, and avoiding predators. In species that live in caves, or the deep sea where vision is useless, the lateral line becomes the primary sensory system. And in the deep sea, where every photon of bioluminescent light is precious and revealing yourself visually could mean becoming prey, the lateral line offers something vision never could: passive detection. You can sense without being sensed. You can track without emitting. You leave no signature except the disturbance you make moving through water, which if you move slowly enough becomes nearly undetectable. Deep-sea fish have taken the lateral line to extremes. Some species have expanded canal systems covering not just the body trunk but the entire head, with branches extending across the skull, around the eyes, along the jaw. The density of neuromasts in these canals far exceeds what you find in shallow water fish. Why? Because in the deep sea, the lateral line isn't supplementing vision, it's replacing it. But the lateral line, as sophisticated as it is, can only detect movement and pressure. It can't detect something that's perfectly still, and it definitely can't detect something buried in sediment. For that you need electroreception. Electroreception, the ability to sense electrical fields, is perhaps the most alien sense to humans. We have no electroreceptive organs. We can't detect bioelectric fields at all. But for many marine animals, particularly sharks, rays, and some deep-sea fish, electroreception is as fundamental as vision is to us. Every living cell generates electrical potential. When muscles contract, neurons fire, or hearts beat. They create tiny electrical fields that propagate through the surrounding medium. In air, these fields dissipate almost instantly, but water, especially seawater with its dissolved salts, conducts electricity remarkably well. Bioelectric fields can extend several feet from their source, and some animals can detect fields as weak as 5 nanovolts per centimetre. That's 5 billionths of a volt. To put that in perspective, if you place 2A batteries 3,000 miles apart in the ocean, a shark could theoretically detect the voltage gradient between them. This extraordinary sensitivity comes from specialised organs called ampullae of Lorenzini, most famous in sharks but present in rays, skates, chimeras, and even some bony fish like paddlefish and sturgeon. Ampullae of Lorenzini appear as small pores on the skin, concentrated mainly on the head. Each pore opens into a canal, sometimes over a foot long, filled with conductive jelly. At the end of each canal sits a cluster of sensory cells, hair cells similar to those in the lateral line but modified to detect voltage instead of pressure. According to research published in scientific reports in 2025 on deep-sea ghost sharks and spookfish, these animals possess extensive ampullary fields covering their heads and snouts, with hundreds or even thousands of individual pores. The sheer density suggests these fish rely heavily on electroreception for hunting, and the strategy makes perfect sense. Most prey organisms, fish, crustaceans, worms, can't stop their hearts, can't prevent their muscles from generating bioelectric fields, can't shut down the electrical activity of their nervous systems, even if they're motionless, even if they're buried under sand, even if they're perfectly camouflaged, they're broadcasting their location electromagnetically. A predator with electroreception can find them anyway. Hammerhead sharks, with their bizarre flattened heads, may have evolved that shape specifically to improve electroreception. The wide, flat head increases the surface area available for ampullae, creating a broader electroreceptive antenna that can scan larger volumes of water and pinpoint prey location with greater precision. Experiments have shown that sharks can detect the heartbeat of a flatfish buried completely under sand. They swim over the seafloor, sweeping their heads back and forth like metal detectors, homing in on the rhythmic electrical pulses of a hidden heart. When they find it, they strike, accurately without ever seeing the prey. But electroreception isn't just for hunting, it's also for navigation. Earth itself generates electromagnetic fields. The planet's magnetic field creates predictable voltage gradients in seawater as ocean currents flow through it. Sharks and rays can detect these gradients. They use them as a navigational compass, orienting themselves relative to Earth's magnetic field, potentially migrating thousands of miles using electromagnetic cues we can't perceive at all. In the deep sea, where there are no visual landmarks, no sunlight to indicate direction, no stars to navigate by, electromagnetic navigation becomes crucial, and electroreception, evolved primarily for detecting prey, doubles as a geomagnetic sense. But both the lateral line and electroreception require external stimuli. They're passive senses, waiting for prey to move or hearts to beat. Some deep-sea creatures have evolved active sensing instead. They don't wait for signals, they create them. This is echolocation, and while we typically associate it with whales and dolphins, some deep-sea fish may use it too, though the evidence remains debated. Echolocation works by emitting sound and listening for echoes. The time delay between emission and return tells you distance. The echo's characteristics tell you size, shape, texture. It's biological sonar, and it works in total darkness. Toothed whales, sperm whales, orcas, dolphins use echolocation constantly, producing clicks and listening to how those clicks bounce off objects. Sperm whales hunt giant squid at depths exceeding 6,000 feet in absolute darkness, using only sound to locate prey the size of school buses in open water. According to research on sperm whale acoustic behaviour, they produce some of the loudest biological sounds ever measured, clicks reaching 230 decibels, louder than a jet engine. These clicks can travel miles through water, and the returning echoes provide enough information for the whale to construct a mental map of its surroundings. But fish don't have the anatomical equipment for echolocation that whales possess. They lack the specialised organs, the massive sound-producing structures, the sophisticated auditory processing centres. Most fish hear reasonably well, but they don't echolocate, except maybe some do. There's tantalising evidence that certain deep-sea fish produce sounds and may use the returning reflections for spatial awareness. Sturgeons, lanternfish, some grenadiers, all have been recorded producing clicks, pops or drumming sounds. Whether these sounds serve echolocation or are simply communication signals remains unclear. What is clear is that deep-sea creatures have evolved to maximise every possible sensory input, because in an environment this hostile, information is survival. And then there's chemical sensing, which might seem mundane compared to electroreception and lateral lines, but in the deep sea becomes extraordinarily refined. Smell and taste, chemoreception, work differently underwater than in air. On land, we inhale air carrying volatile molecules, and olfactory receptors in our noses detect them. Taste requires direct contact, molecules dissolved in saliva touching taste buds, but underwater, everything is already dissolved. The distinction between smell and taste blurs. Fish have chemoreceptors distributed across their bodies, in nostrils, in the mouth, on fins, even on barbels, whisker-like appendages extending from the head. Deep-sea fish often have enormous olfactory organs relative to their body size. Some species have olfactory bulbs, the brain region processing smell, that dwarf the rest of the brain. Smell becomes the dominant sense. Why? Because chemical signals persist. A drop of blood, a trace of amino acids from decaying flesh, molecules sloughed off by living tissue. These substances diffuse slowly through water, creating plumes that can extend hundreds of meters. A fish with sensitive chemoreceptors can follow those plumes to their source, tracking prey or scavenging opportunities across vast distances. Hagfish, primitive, eel-like scavengers that inhabit the deep-sea floor, are perhaps the best example. Hagfish are nearly blind. They have no lateral line system. Their hearing is poor, but they have exquisitely sensitive chemoreceptors that can detect a single drop of fish oil diluted into millions of gallons of water. When a whale carcass falls to the deep sea floor, whale fall, one of the most nutrient-rich events in the deep ocean, hagfish can smell it from miles away. They converge on the carcass within hours, sometimes arriving in such numbers that the seafloor around the whale becomes carpeted with writhing bodies, all feeding simultaneously. They don't need to see the food. They smell their way to it through kilometers of dark water. But chemoreception isn't just for finding food, it's also for reproduction. Many deep-sea species exist at such low population densities that encounters between males and females are vanishingly rare. In the largest habitat on Earth, with individuals scattered across millions of cubic miles of water, finding a mate is statistically almost impossible, so females broadcast. They release pheromones, chemical signals that advertise reproductive readiness. Males, sometimes equipped with larger olfactory organs than females, detect these pheromones and follow the chemical gradient to its source. In some deep-sea anglerfishes, this strategy reaches an extreme. Males are tiny, sometimes 1/10 the size of females, and their entire existence revolves around finding a female. Once they detect a female's pheromones, they pursue relentlessly. Upon contact, the male bites the female and never lets go. His tissues fuse with hers. His circulatory system connects to hers. Over time, he degenerates, losing eyes, fins, most internal organs, until he becomes essentially a parasitic sperm-producing appendage attached to the female's body. She carries him with her, and when she's ready to spawn, his sperm is right there, permanently available. It's sexual parasitism enabled by chemoreception. The male found the female in the vastness of the deep ocean by smell alone, then ensured he'd never lose her. But even with all these senses, lateral lines detecting pressure waves, ampullae sensing bioelectric fields, chemoreceptors tracking molecular trails. There's one sensory challenge the deep sea creates that seems insurmountable. How do you use vision in a place where light doesn't exist? You'd think deep sea creatures would be blind. Many are. But others have evolved eyes more sophisticated, more sensitive, more specialized than almost anything found in shallow water. Deep sea fish eyes come in bizarre forms. Some species have tubular eyes shaped like binoculars, pointed upward, maximizing light collection from the faint bioluminescence above. The barreleye fish we discussed earlier exemplifies this. Eyes embedded in a transparent head, rotating inside their sockets, collecting every photon available. Other species have enormous eyes relative to their body size. The giant squid has eyes the size of dinner plates, the largest eyes in the animal kingdom. Not because it lives in well-lit water, but because massive lenses can collect more of the scarce light available at depth. And the retinas of deep-sea fish are packed with rod cells, the photoreceptors specialized for low-light vision. Some species have retinas composed almost entirely of rods, with no cone cells at all. They've sacrificed color vision entirely in exchange for maximum sensitivity to dim light. According to research on deep-sea fish vision, some species have evolved rhodopsin, the light-sensitive pigment in rod cells, with altered spectral sensitivities tuned specifically to detect bioluminescence at the wavelengths most common in their habitat. Their eyes aren't just sensitive, they're precisely calibrated to see the kind of light their prey, predators, and potential mates produce. But perhaps the strangest visual adaptation is found in species with multiple visual pigments in the same retina, not for color vision, but for detecting different intensities of bioluminescence, or distinguishing bioluminescent light from residual sunlight at twilight depths. The stoplight loosejaw dragonfish, which produces red bioluminescence, has eyes that can see red light. Rare in deep-sea fish, since red wavelengths don't penetrate deep water. This gives the dragonfish a private channel. It can illuminate prey with red light that only it can see, hunting with a spotlight invisible to everyone else. And then there are fish with accessory retinas, extra light collecting structures behind the main retina that reflect light back through the photoreceptors for a second pass, doubling the chance of photon detection. It's biological image amplification, like night vision goggles built into the eye. What all these adaptations reveal is that the deep sea isn't truly dark. It's dim, extremely dim. But dim enough that evolution could work with it, refining vision to absurd extremes, extracting information from almost nothing. But when vision fails, when light is truly absent, deep-sea creatures still navigate, still find food, still reproduce. Because they've built sensory systems that don't depend on light at all. They sense pressure waves through lateral lines. They detect heartbeats through electroreception. They smell molecules through chemoreceptors. They feel vibrations through mechanosensors distributed across their skin. They use statocysts, balance organs filled with calcium carbonate crystals, to maintain orientation in three-dimensional space. They've turned the ocean into a readable environment without needing to see it. And what this means is profound. These creatures aren't handicapped by darkness. They're not limited by the absence of light. They've simply moved to different information channels. They're perceiving a version of reality we can't access, building mental maps of their world from electrical fields, pressure gradients, and chemical trails. To them, the deep ocean isn't a sensory void. It's dense with information. It's a landscape as rich and detailed as any rainforest or coral reef, just written in a language we don't speak. And that raises a final, unsettling question. What else is out there that we can't detect? Not because it doesn't exist, but because our senses aren't tuned to perceive it. The deep ocean taught evolution that the senses we take for granted, vision, hearing, smell as we experience them, are just a narrow slice of available reality. There are fields, gradients, vibrations, signatures all around us that we're blind to. Deep-sea creatures see them, feel them, read them like text, and they remind us that reality isn't what exists, it's what we're equipped to perceive, which means the ocean might be even stranger than we think. Because if our technology can reveal worlds invisible to us, what might exist that even our technology can't detect yet? The deep ocean keeps its secrets, not because they're hidden, but because we're still learning how to look. There's a woodlouse, a pillbug, the little armoured crustacean you find under rocks in your garden, living in the deep sea that can grow to 30 inches long. 30 inches, that's the length of a house cat. It's called the giant isopod, Bathynomus giganteus, and it's one of the most dramatic examples of a phenomenon called deep-sea gigantism, the tendency for creatures living in the abyss to grow far larger than their shallow water or terrestrial relatives. A typical woodlouse is maybe half an inch long. The giant isopod is 60 times that size. 60 times? That's not adaptation, that's reinvention, and it's not alone. The giant squid reaches lengths over 40 feet. The colossal squid, even larger, may exceed 45 feet and weigh over 1,500 pounds. Japanese spider crabs have leg spans reaching 12 feet. Giant amphipods, shrimp-like crustaceans, can grow to the size of a human hand, when their shallow water cousins are barely a millimetre long. Giant tube worms stretch nearly 8 feet. Giant oarfish reach 30 feet. The list goes on. Why? What is it about the deep ocean that doesn't just allow gigantism, but seems to encourage it? The answer lies in a combination of physics, metabolism, and evolutionary pressure that makes the deep sea one of the few places on Earth where bigger is almost always better. Start with buoyancy. On land, size is constrained by gravity. The largest land animals, elephants, giraffes, are at the physical limits of what bone and muscle can support. Go bigger, and skeletal systems fail. Legs break. Hearts struggle to pump blood against gravity. Movement becomes impossible. But water changes everything. Water provides buoyancy. An organism's weight is counteracted by the water it displaces. In the ocean, you don't need massive skeletons to support body weight. You don't need powerful legs to stand. Structural support comes from water pressure equalizing inside and outside the body. This is why blue whales, the largest animals ever to exist, larger than any dinosaur, can only exist in water. On land, a blue whale would collapse under its own weight, its organs crushed, its skeleton inadequate. But in water, it floats effortlessly, 200 tons moving with grace through three-dimensional space. The same principle applies to deep-sea invertebrates. A giant isopod doesn't need thick armour or powerful muscles to support its enormous body. The water does that. Its exoskeleton needs only enough rigidity to protect organs and provide attachment points for muscles. The rest is gelatinous tissue, minimally structured, held in shape by internal and external pressure equilibrium. But buoyancy alone doesn't explain gigantism. Shallow water species have buoyancy too, and they stay small, so there must be other factors. Enter Kleiber's law, one of the most fundamental scaling principles in biology. Kleiber's law states that an animal's metabolic rate doesn't scale linearly with mass. It scales to roughly the 3/4 power of mass. In simpler terms, if you double an animal's weight, its metabolic rate doesn't double. It only increases by about 75%. Why? It comes down to surface area, volume, and the geometry of circulatory and respiratory systems. Metabolism generates heat. Heat must be dissipated through the skin. But surface area increases as the square of linear dimensions, while volume increases as the cube. Bigger animals have proportionally less surface area per unit mass, so they lose heat more slowly. To avoid overheating, they've evolved lower mass-specific metabolic rates. Additionally, the circulatory system, which delivers oxygen and nutrients, operates more efficiently at larger scales. Blood vessels, airways, and transport systems don't need to scale one-to-one with body size. A fractal branching network can service a large body with proportionally less metabolic overhead than servicing many small bodies with the same total mass. The upshot: large animals are metabolically more efficient. They need less food per gram of body weight than small animals. According to Kleiber's law, a cat weighing 100 times more than a mouse requires only about 32 times as many calories. Not 100 times, just 32. In an environment where food is abundant, this doesn't matter much. But in the deep sea, where food is desperately scarce, metabolic efficiency becomes survival. The deep ocean is a desert. Sunlight doesn't penetrate, photosynthesis doesn't occur. The only food comes from above. Marine snow, the slow rain of organic debris drifting down from surface waters, dead plankton, fecal pellets, molted exoskeletons, fragments of jellyfish. Occasionally, a large carcass, a whale, a shark, a tuna, falls from above, but these events are rare, unpredictable, separated by years or decades. For a deep-sea scavenger, meals are sparse and sporadic. You might go months without encountering food. When you do find it, you need to consume as much as possible, storing it for the long famine ahead. This is where Kleiber's law becomes crucial. A large animal can survive longer between meals because it burns less energy per gram of tissue. It can store more reserves relative to its maintenance costs, and when food appears, it can consume more in a single feeding event. Giant isopods exemplify this. According to research on their feeding behaviour, they can gorge themselves to the point of near immobility, distending their bodies until they can barely move, and then they wait. Studies have shown that giant isopods in captivity can survive over 5 years without food. 5 years. That's not starvation resistance in the sense we usually think of it. That's a fundamentally different metabolic strategy. One where the baseline energy expenditure is so low that stored reserves can last half a decade. And it's only possible at large body sizes. A tiny isopod, with its higher mass-specific metabolic rate, would starve in months. But scale up to 30 inches, and suddenly 5 years of fasting becomes feasible. So buoyancy enables large size, and Kleiber's law makes large size advantageous in food-scarce environments. But there's a third factor: temperature. This is where Bergmann's rule comes in. The observation that animals in colder climates tend to be larger than their warm climate relatives. The principle is straightforward. Larger animals have lower surface area to volume ratios, so they lose heat more slowly. For warm-blooded animals trying to maintain body temperature, larger size conserves warmth, but the deep ocean is cold, near freezing, and most deep-sea creatures are cold-blooded. Their body temperature matches the water, so Bergmann's rule shouldn't apply, right? Except it does, for a different reason. Cold water holds more dissolved oxygen than warm water, and according to a 1999 study published in Biological Bulletin, oxygen availability may directly influence maximum body size in deep-sea organisms. The study found that in amphipods, small crustaceans, maximum size correlated strongly with dissolved oxygen levels. Species living in cold, oxygen-rich, deep water grew significantly larger than species in warmer, oxygen-depleted zones. The reason? Larger bodies require more oxygen to sustain, but they also have more efficient oxygen delivery systems. In oxygen-rich environments, the benefits of large size, metabolic efficiency, lower predation risk, outweigh the costs, higher absolute oxygen demand. In oxygen-poor environments, the calculus reverses. Cold deep sea water, saturated with oxygen, removes one of the main constraints on size, and evolution responds by pushing body size upward. But predation also plays a role. Predation pressure in the deep sea is dramatically lower than in shallow water. According to studies on deep-sea brachiopods, predation rates at abyssal depths are nearly an order of magnitude lower than in coastal ecosystems. Why? Fewer predators, lower population densities, slower movement speeds, less frequent encounters. In shallow water, being small helps you hide, being fast helps you escape. But in the deep sea, where encounters are rare and predators are often ambush hunters, rather than pursuit predators, size becomes a defense. A giant isopod is too large for most deep-sea predators to handle. Its thick exoskeleton is difficult to penetrate. Even if attacked, its size means it can survive injuries that would kill smaller animals. So reduced predation pressure removes another constraint on size, allowing evolution to favour gigantism for metabolic reasons without suffering predation costs. Put it all together. Buoyancy enables large size. Kleiber's law makes it metabolically advantageous. Cold temperatures increase oxygen availability. Reduced predation pressure eliminates one of the main costs. Food scarcity makes efficiency paramount. The result? Gigantism becomes the dominant strategy, but it's not universal. Deep-sea gigantism applies to specific groups: crustaceans, cephalopods, cnidarians, some eels. But not all deep-sea creatures are giants. In fact, the smallest organisms in the deep sea, meofauna, creatures smaller than 1 millimeter, show the opposite trend. They get smaller with depth, not larger. Why the difference? It comes down to lifestyle and feeding strategy. Gigantism works for scavengers and predators, organisms that need to travel, search, and consume large prey items when found. For them, metabolic efficiency and the ability to fast for long periods are critical. But for filter feeders, detritus feeders, and animals that glean organic particles from sediment, small size is advantageous. Smaller bodies have higher surface area to volume ratios, which makes passive feeding more efficient. They can extract more nutrients per unit of body mass from the thin film of organic material coating surfaces. So the deep sea doesn't impose one single size solution. It creates selective pressures that favour different sizes depending on how you make a living. But for the giants, the creatures that capture our imagination, the common thread is patience. Deep-sea giants move slowly. The giant squid, despite its size, is thought to be a relatively sluggish swimmer. The giant isopod crawls across the seafloor at a glacial pace. Greenland sharks, which can grow to 20 feet and live over 400 years, swim at less than a mile per hour. They're among the slowest moving sharks in the ocean. This isn't weakness. It's strategic. Movement requires energy. In an environment where energy is precious, you conserve it. You wait. You ambush. You scavenge. You do everything as slowly as possible, expending the minimum necessary to survive. And gigantism supports this lifestyle. Larger bodies store more energy. They can afford to wait longer. They can survive the lean times between windfalls. But there's a cost. Giant deep-sea creatures reproduce slowly. They grow slowly. They reach sexual maturity late. Their populations are small, their numbers scattered across vast areas. This makes them vulnerable. When deep-sea fishing or mining disrupts their habitat, recovery takes decades or centuries. A giant isopod that can live 5 years without food might take 10 years to reach maturity. If you remove a significant portion of the population, replacement rates are agonizingly slow. The giants of the deep have traded speed for endurance, rapid reproduction for longevity. They've optimized for a stable, unchanging environment where patience is rewarded and haste is wasteful. But that strategy assumes the environment stays stable, and increasingly human activity is disrupting the deep sea. Trawling, mining, pollution, climate change, altering temperature and oxygen levels. The giants evolved over millions of years to thrive in stillness. Now that stillness is breaking, and the same adaptations that made them successful Slow metabolism, low reproductive rates, extreme specialization make them fragile when conditions change quickly. The deep ocean taught us that life can grow enormous when freed from gravity and optimized for scarcity. But it's also teaching us that giants, despite their size, can be surprisingly fragile. Which brings us to the final paradox of deep-sea life. How do you reproduce when finding a mate is statistically nearly impossible, when individuals are scattered across millions of cubic miles, when your entire species might number in the hundreds or thousands across an entire ocean basin? The answer involves some of the strangest reproductive strategies evolution has ever produced. In 1925, a British ichthyologist named Charles Tate Regan was examining a female anglerfish specimen when he noticed something odd. There were small, parasitic-looking growths attached to her belly. He assumed they were parasites, perhaps some kind of deep-sea barnacle or worm. They weren't. They were males, fused males that had bitten into the female's tissue, released an enzyme that dissolved the boundary between their bodies, and permanently attached themselves. Their circulatory systems had merged. Their tissues had fused. They'd given up independence, given up feeding, given up almost everything except their reproductive organs. They'd become living sperm banks, carried through the abyss by females who would fertilize eggs on demand. This is sexual parasitism, and it represents one of the most extreme solutions to the fundamental problem facing all deep-sea life. In a habitat so vast and so empty, how do you ever find a mate? Consider the statistics. The deep ocean is the largest habitat on Earth, roughly 1.3 billion cubic kilometers of water. Most deep-sea species have small population sizes, often numbering in the thousands or tens of thousands, spread across entire ocean basins. Individual organisms can go years without encountering another member of their species. For animals that reproduce sexually, this creates an existential crisis. Sexual reproduction requires two individuals to find each other in the right place, at the right time, when both are reproductively mature. In a coral reef, that's challenging but manageable. In the abyss, it's statistically nearly impossible. Evolution has responded with strategies so bizarre they seem like science fiction. Let's start with those anglerfish males. In many deep-sea anglerfish species, particularly those in the suborder Cerathiidae, males are dramatically smaller than females, sometimes 1/10 the size. They have enormous eyes relative to their body, which makes sense. Their entire purpose is finding a female in the dark. Once a male hatches, he has a limited window. He doesn't feed. He can't. His digestive system is vestigial, sometimes entirely absent. His olfactory organs, however, are massively developed. He can detect pheromones, chemical signals released by females, across vast distances, following concentration gradients through the water like a bloodhound following a scent trail. When he finally finds a female, he bites into her skin, usually near the belly or flanks. His mouth releases enzymes that break down tissue boundaries. Within days, the circulatory systems merge. His blood vessels connect to hers. Nutrients from her bloodstream keep him alive. His body degenerates. Eyes shrink, fins atrophy, internal organs disappear. What remains is essentially a pair of gonads with a blood supply, permanently grafted onto the female. When she's ready to spawn, hormones in her bloodstream trigger sperm release. She controls the timing. He's along for the ride. Some female anglerfish carry multiple males, sometimes 3 or 4 fused individuals scattered across her body. She's created her own travelling harem, ensuring she'll never need to search for a mate again. It's parasitism. It's also partnership. Neither sex could reproduce alone in the abyss. The male gets transportation, nutrition, and guaranteed mating opportunities. The female gets on-demand fertilization, without the nearly impossible task of finding a mate when she needs one. It's disturbing from a human perspective. From an evolutionary perspective, it's brilliant. But sexual parasitism is only one solution. Other species use different strategies, equally extreme. Take synchronous spawning. In shallow water coral reefs, we see mass spawning events where hundreds of species release eggs and sperm simultaneously on the same night, usually coordinated with lunar cycles. It's spectacular, turning the water into a blizzard of gametes. The deep sea has no moon, No visible lunar cycle, no seasons in the traditional sense. Time in the abyss is almost meaningless. Yet some deep-sea species still manage to synchronize reproduction. Research published in Deep Sea Research in 2019 by biologist Mance Preed and colleagues found evidence of synchronized spawning in deep-sea fish populations based on gonad development patterns across specimens collected at similar times. How do they coordinate without environmental cues? Pheromones again. Chemical signals that don't just attract mates but can trigger reproductive readiness in other individuals who detect them. One fish reaches sexual maturity and begins releasing spawning pheromones. Others detect these chemicals and their own reproductive systems respond, accelerating egg or sperm development. The effect cascades through the local population. Within a few weeks, most mature individuals in the area are ready to spawn simultaneously. When spawning begins, the release of massive amounts of pheromones triggers a chain reaction. Everyone spawns at once, maximizing the chances that eggs and sperm find each other in the water column. It's chemical coordination, a form of distributed decision-making. No single individual controls the timing. The population decides collectively through molecular communication. Then there's the strategy of producing enormous numbers of offspring and hoping a few survive. Deep-sea fish like the ocean sunfish, mola mola, can produce up to 300 million eggs in a single spawning event. 300 million. It's the highest fecundity of any vertebrate on Earth. Why so many? Because mortality is astronomical. Deep-sea larvae are tiny, fragile, and drift in the water column for weeks or months. They're eaten by everything. Predation rates exceed 99.9%. Of those 300 million eggs, maybe a few dozen will survive to adulthood. It's the opposite strategy from mammals or birds, which invest heavily in a small number of offspring and provide parental care. Deep-sea species go for volume, broadcast spawning, release millions of gametes into the water, let currents mix them, and hope random chance brings enough together to maintain the population. There's no care, no protection, the adults spawn and disappear, the offspring are on their own from the moment of fertilization. This works only because deep-sea adults are long-lived. If a fish lives 80-100 years and spawns multiple times, producing millions of eggs each time, the cumulative odds shift in favor of successful reproduction. But it's a brutal strategy. Most individuals will never reproduce. Most lineages die out. Only a tiny fraction succeed. Compare this to deep-sea octopuses, which take the opposite approach. Female deep-sea octopuses in the genus Granellodon, particularly Granellodon boreopacifica, have been observed guarding their eggs for over 4 years. 4 years. That's the longest egg-brooding period of any known animal. In 2014, researchers from the Monterey Bay Aquarium Research Institute published observations of a single female G. boreopacifica at 4,600 feet depth, who remained in the exact same spot for 53 months, never leaving her clutch of roughly 160 eggs. She didn't feed. She constantly circulated water over the eggs with her arms, keeping them clean and oxygenated. By the end, she was emaciated, skin pale and torn, barely moving. When the eggs finally hatched, she died. She'd invested everything, literally her entire body's energy reserves, into ensuring those 160 offspring had the best possible start. No feeding, no movement, no recovery, just protection, and then death. Why such extreme maternal investment? Because in the deep sea, larvae survival rates are abysmal. Surface-dwelling octopuses produce tens of thousands of eggs and provide minimal care. Their larvae are planktonic, drifting with currents, feeding themselves. Most die, but enough survive because conditions are relatively rich. Deep-sea octopuses can't afford that strategy. Food is too scarce. Larval development takes too long. If you release planktonic larvae into the abyss, they'll starve before reaching independence. So deep-sea octopuses produce fewer, larger eggs with more yolk. Each one is a survival package, a miniature food supply. And the mother guards them until they're developed enough to hunt on their own. It's the only way to give them a fighting chance. She dies so they can live. There's something almost unbearable about that image. A single female in the darkness, starving slowly, arms moving gently over eggs she'll never see hatch. It's devotion at the cellular level. Evolution has optimized her biochemistry to shut down everything except the imperative to protect. reproduction in the deep sea isn't about pleasure or bonding or elaborate courtship. It's logistics. It's solving an impossible equation. How do you ensure genetic continuity when individuals are scattered across millions of square miles, when energy is scarce, when meeting a mate might happen once in a lifetime or never? The solutions are as varied as the species. Some fuse permanently, some coordinate chemically, some spawn millions of offspring, some dedicate years to a handful, but they all work. They've worked for millions of years. The deep ocean is full of life, reproducing, continuing, persisting, against odds that would seem to make it impossible, and that tells us something profound about evolution. It doesn't aim for elegance or efficiency or beauty. It aims for whatever works, and if what works is males permanently grafted to females, or mothers starving to death over eggs, or fish broadcasting 300 million gametes into the void, then that's what evolution produces. Life doesn't care how strange it looks to us. It cares about continuation. Which brings us to the final piece of the puzzle, time. Because all of these adaptations, the slow metabolisms, the extreme reproductive strategies, the gigantic bodies and patient hunting, evolved over timescales we can barely comprehend. The deep ocean has been stable for hundreds of millions of years. That stability allowed specialization at a level impossible in more dynamic environments. Species could fine-tune every aspect of their biology to extract maximum advantage from an unchanging habitat. But that stability is ending, and the creatures adapted to thrive in stillness are now facing change they're not equipped to handle. For hundreds of millions of years, the deep ocean has been the most stable environment on Earth. Temperature barely fluctuates. Pressure remains constant. Seasons don't exist. Light never penetrates. The rhythms of surface life, daylight, weather, tides, fade into irrelevance at depth. This stability allowed life to specialize in ways impossible elsewhere. A giant isopod can afford a metabolism so slow it goes years without eating because its environment won't suddenly change. An anglerfish male can afford to permanently fuse to a female because resource predictability means both will survive. A deep-sea octopus can afford to starve for 4 years guarding eggs because her offspring will hatch into a world that looks exactly like the one she knew. Evolution thrives on stability. Give a lineage millions of years in unchanging conditions, and it will optimize every aspect of its biology to extract maximum advantage from that specific set of circumstances. It will shed unnecessary features. It will fine-tune metabolic pathways. It will become exquisitely, beautifully adapted. But specialization is a double-edged sword. The more finely tuned an organism becomes to a specific environment, the more vulnerable it is to change. And the deep ocean is changing, not slowly across evolutionary timescales that allow adaptation, rapidly, within decades, within years, faster than deep-sea life can respond. The changes are coming from multiple directions at once, each compounding the others, and most of them trace back to human activity. Start with temperature. Deep-sea temperatures, long stable, are now rising as climate change warms surface waters and heat slowly diffuses downward. The changes are small, fractions of a degree per decade, but for organisms adapted to extreme stability, even small changes matter. Temperature governs metabolism. Warmer water means higher metabolic rates, which means organisms need more oxygen and more food to sustain themselves. But the deep sea doesn't have more food to give. Production is already at the limit of what can sink from the surface. If metabolic demands increase while food supply remains constant or decreases, organisms starve. Some species might adapt by moving deeper, following their preferred temperature range as isotherms shift. But many deep-sea species are already near the limits of pressure tolerance. There's nowhere colder to go. The abyss ends at the seafloor. Then there's oxygen. The ocean is losing oxygen. As surface temperatures rise, water holds less dissolved gas. Warm water is less dense, which strengthens stratification, the layering of the ocean into distinct temperature zones that don't mix well. When stratification increases, less oxygen-rich surface water circulates to depth. Oxygen minimum zones are expanding both horizontally and vertically, pushing into regions that were previously well oxygenated. For species already living at the edge of hypoxia, this is catastrophic. There's no adaptation fast enough to compensate for suffocation. Animals that spent millions of years evolving to tolerate low oxygen suddenly find themselves in conditions where even their extreme adaptations aren't enough. Then there's acidification. The ocean absorbs about a quarter of the carbon dioxide humans emit into the atmosphere. When CO2 dissolves in seawater, it forms carbonic acid, lowering pH. The surface ocean has already acidified by about 30% since the Industrial Revolution began. That acidification is now spreading to depth. Many deep-sea organisms have shells or skeletons made of calcium carbonate. Corals, mollusks, echinoderms, certain plankton. In acidic water, calcium carbonate dissolves, not quickly, but inevitably. The chemistry shifts against them. Building and maintaining shells becomes harder. Energy that should go toward growth or reproduction gets diverted to structural maintenance. Some deep-sea species might not survive at all, as pH drops below thresholds their biochemistry can tolerate. These are the indirect impacts, the creeping changes in baseline conditions that alter the fundamental habitability of the deep ocean. They're slow, hard to detect, and nearly impossible to reverse once set in motion. But there are also direct impacts, physical destruction, and these are happening now at scales that dwarf anything in the deep ocean's recent history. Bottom trawling. Bottom trawling is a fishing method where weighted nets, some with metal doors weighing several tons and foot ropes studded with steel balls, are dragged across the seafloor to catch fish. The nets scrape, crush and bulldoze everything in their path. On soft sediment bottoms, mud and sand, trawling kills in fauna, resuspends sediment and disrupts feeding and reproductive behaviours. Studies in the Mediterranean found that chronically trawled areas showed organic matter content reduced by up to 52%, meiofauna abundance down 80%, and biodiversity reduced by 50%. But the worst damage occurs on hard substrates, seamounts, ridges, cold water coral reefs. These are biodiversity hotspots, the rainforests of the deep. Deep-sea corals can be hundreds to thousands of years old, with some dated at over 4,000 years. They grow millimetres per year, building massive reef structures that provide habitat for thousands of species. A single trawl pass destroys them in seconds. Research on seamounts off Tasmania found that even 14 years after trawling stopped, there were no signs of recovery. The community structure remained degraded, resembling actively trawled sites. 14 years for ecosystems that took millennia to develop. The United Nations World Ocean Assessment found that bottom trawling has caused widespread, long-term destruction globally, and that recovery may take centuries to millennia. We are turning portions of the deep sea floor into deserts. Not metaphorically, literally. Areas that once supported diverse communities are now barren, dominated by a handful of opportunistic species adapted to disturbance. And trawling isn't slowing down. As coastal fisheries collapse from overfishing, industrial fleets push deeper, targeting slow-growing species like orange ruffy, which can live over 200 years and take decades to reach reproductive maturity. These fish evolved life histories optimized for stability, slow growth, late maturity, long lifespans, strategies that work when mortality is low and predictable, but they can't withstand industrial harvest. Populations crash within years of exploitation beginning, and recovery, if it happens at all, takes generations. We're mining fish, not fishing sustainably, extracting accumulated biomass and leaving behind degraded ecosystems. But if bottom trawling is bad, what's being proposed next is potentially catastrophic. Deep sea mining. The seafloor holds vast mineral deposits, polymetallic nodules rich in cobalt, nickel, copper and rare earth elements. These nodules formed over millions of years as metals precipitated from seawater and accumulated layer by layer on the ocean floor. They grow at rates of millimetres per million years. They also serve as habitat. The rough surfaces provide attachment sites for sponges, corals, anemones. The nodules create structural complexity in otherwise flat abyssal plains. Studies show that the majority of biodiversity in nodule fields lives directly on the nodules themselves. Deep-sea mining would vacuum these nodules up, along with everything attached to them, along with the top few inches of sediment. The mining vehicles would churn the seafloor, creating massive sediment plumes that drift for kilometres, smothering organisms downwind. The wastewater from mineral processing, a toxic slurry of sediment, crushed animals and chemicals, would be dumped back into the ocean, creating discharge plumes that spread over vast areas. Mining would generate intense noise and light pollution in environments that are naturally silent and dark. Sound and light could disrupt communication, reproduction, and feeding behaviours in ways we can't yet predict and recovery. A study published in Nature in 2025 examined a site where experimental nodule collection occurred 40 years earlier. 4 decades later, biological communities had not recovered. Many species were still absent. The seafloor showed persistent physical changes 40 years and no recovery. For an ecosystem that evolved over tens of millions of years, scientists warn that biodiversity loss from deep-sea mining would be inevitable and most likely irreversible. Species could go extinct before we even discover they exist. Ecosystem services, carbon sequestration, nutrient cycling, fisheries support could collapse. Despite these warnings, the International Seabed Authority has been negotiating regulations to allow commercial mining to begin, potentially by 2025. Companies are already testing equipment. Norway announced in 2023 that it would proceed with exploration of deep-sea mining in its waters, becoming one of the first nations to authorize such activity. The rush is driven by demand for batteries, electric vehicles, renewable energy storage, electronics. Proponents argue that deep-sea mining avoids the environmental and human rights problems of terrestrial mining. No deforestation, no displaced communities, no child labour in cobalt mines. But economic analyses suggest deep-sea mining would generate minimal benefits while imposing massive environmental costs. A study by Planet Tracker estimated that deep-sea mining could destroy over $500 billion in natural capital and corporate value. Small island nations that depend on tuna fisheries could see catches drop by 10% or more if mining disrupts marine ecosystems in targeted regions. The people with the least responsibility for climate change would bear disproportionate harm from a supposed solution to it. And there are alternatives: recycling, material substitution, more efficient use of existing resources, circular economy models that eliminate the need for virgin mineral extraction entirely. Several countries, Chile, Palau, Fiji, Vanuatu, France, have called for moratoriums on deep-sea mining until comprehensive environmental assessments can be completed. They recognise what should be obvious: we don't know enough to do this safely, and once the damage is done, it can't be undone. But the pressure continues. The International Seabed Authority moves forward with regulations, test equipment is deployed, the clock is ticking, and the deep ocean, which has no voice, no constituency, no political power, faces industrialization before we've even finished exploring it. We've mapped more of Mars than we have of our own seafloor. We've named more craters on the moon than seamounts in the deep ocean. And we're preparing to mine ecosystems we don't understand, destroying habitats we've barely seen, erasing species we haven't discovered. The irony is suffocating. We look to the stars for alien life while ignoring the aliens in our own ocean. We spend billions searching for microbes on Mars while bulldozing coral reefs that took millennia to grow. We fantasize about terraforming other planets while rendering large portions of our own uninhabitable. The deep ocean survived 5 mass extinctions. It persisted through ice ages, through volcanic cataclysms, through asteroid impacts. It adapted to every change the planet threw at it across 3.5 billion years. But it never faced anything like this. It never faced a single species capable of altering global chemistry, warming the entire ocean, and physically destroying habitat at industrial scales within decades. The organisms of the deep sea evolved patience. They evolved to wait, to endure, to outlast. Their entire biology is built on the assumption that stability will continue, that time stretches on, that tomorrow looks like yesterday. That assumption is breaking. And when an ecosystem optimized for stability faces rapid change, the result isn't adaptation, it's collapse. The giant isopods, the anglerfish, the deep-sea octopuses, the coral forests, they're not equipped for this. Their slow metabolisms can't speed up. Their low reproductive rates can't increase. Their specialized biochemistry can't pivot. Evolution doesn't work that way. Adaptation requires variation, selection, and time. Deep-sea species have variation, though often less than shallow water relatives due to stable conditions and small population sizes. They're certainly under selection pressure now, but they don't have time. Recovery from disturbance, when it happens at all, takes decades to centuries. Extinction happens in years. We're forcing deep-sea life to play a game they can't win, under rules evolution never prepared them for, on timescales that make adaptation impossible. And the tragedy is, it's not even necessary. We don't need to mine the deep sea. We don't need to trawl every seamount. We could choose differently. We have the technology, the knowledge, and the resources to meet human needs without destroying the last great wilderness on Earth. But choice requires will, and will requires caring, and it's hard to care about what you can't see. The deep ocean is out of sight. Its inhabitants are strange, unfamiliar, sometimes unsettling. They don't have big eyes or fur. They don't trigger mammalian empathy circuits. A giant isopod doesn't inspire the same protective instinct as a panda. But they're no less alive, no less the product of billions of years of evolution, no less worthy of existence, and they might be teaching us something we desperately need to learn: that life doesn't require familiarity to be valuable, that intelligence doesn't need brains to be real, that Earth contains worlds as alien as any we'll find in space. The ocean didn't evolve life, it invented another one. A parallel experiment in what biology can become when freed from terrestrial constraints, when pressure replaces gravity, when darkness becomes the canvas, and chemistry becomes language. For 4 billion years, life found ways to persist. In boiling vents and frozen plains, in acid and vacuum and crushing dark. It adapted, diversified, filled every niche, turned every challenge into opportunity. The deep ocean represents the final chapter of that story on Earth. The last frontier where evolution still writes new possibilities, where undiscovered species outnumber known ones, where the fundamental rules of life are being tested in ways we're only beginning to understand. And we're about to close the book, not through malice, through indifference, through the banal logic of extraction that sees resources instead of ecosystems, commodities instead of communities, profit instead of irreplaceable complexity. The deep ocean can't speak for itself. Its inhabitants can't protest, can't vote, can't appeal to our reason or compassion. They exist in silence, in darkness, beyond our sensory world. Which means the responsibility falls to us. To recognize what we're losing before it's gone. To understand that alien doesn't mean expendable. To accept that some places should remain beyond the reach of industry, not because they lack value, but because their value transcends extraction. The creatures of the deep sea evolved every strategy imaginable to survive in an impossible place. They solved problems physics said couldn't be solved. They built communities in emptiness, found food in starvation, reproduced against impossible odds. They survived everything Earth could throw at them, everything except us. We began this journey asking how life could be so different beneath the waves, how evolution could produce organisms so alien, so strange, so unlike anything on land. The answer is simple. The ocean isn't like land. It never was. It's a three-dimensional volume where gravity weakens, where pressure dominates, where light dies and chemistry speaks. It's a physical regime so different from the terrestrial surface that life had to reinvent itself to exist there. And in that reinvention, it created wonders. Bioluminescence that turns darkness into language. Pressure-adapted proteins that function at depths that would crush steel. Metabolism so efficient they can extract energy from near nothing. Reproductive strategies that solve the mathematics of loneliness in the largest habitat on Earth. These adaptations aren't just curiosities. They're proof that life is more flexible, more creative, more resilient than we imagined. That the rules we think govern biology are just the rules governing terrestrial biology. That the universe of possible life forms is vastly larger than the tiny subset we see on land. The deep ocean shows us what life becomes when the constraints change, and in doing so, it hints at what we might find elsewhere. On Europa's ice-covered ocean, on Enceladus, on worlds we haven't discovered yet. But first, we have to protect the alien world we already have. Because if we can't preserve the biodiversity on our own planet, in our own ocean, we have no business reaching for others. The deep sea is still here, Still alive, still evolving. The giant isopods still scavenge, the anglerfish still hunt, the octopuses still guard their eggs in the dark, but the window is closing, the stability is ending, and the creatures who evolved to endure are running out of time to adapt. The choice is ours. We can be the generation that industrialized the abyss and erased ecosystems before we understood them, or we can be the generation that recognized what we were about to lose and chose differently. We can see the deep ocean as alien, yes, but also as kin, as fellow travelers through deep time, as proof that Earth is stranger and richer than any planet we'll visit in our lifetimes. The ocean didn't just evolve life, it evolved possibilities, and every species we lose is a possibility that will never exist again. Every ecosystem we destroy is a question we'll never get to ask. We started by marveling at how different life is beneath the waves. We end by realizing that different doesn't mean separate, that the deep ocean, for all its strangeness, is still Earth, still our biosphere, still part of the planetary web that sustains all life, including ours. And if we're willing to destroy it simply because we can't see it, what does that say about how we'll treat everything else invisible to us? Future generations, other species, the thin envelope of atmosphere and ocean that makes this rock habitable? The deep sea asks nothing of us except what all life asks: room to exist, time to continue. The dignity of not being erased by indifference. It's a small request, and it's the least we owe the creatures who proved that Earth is more alien, more beautiful, and more full of possibility than we ever imagined. They've been evolving in the dark for hundreds of millions of years. The least we can do is let them continue.